longer than usual. Please listen to me very carefully. So this is the second part of gastrointestinal system. As you would remember, last week we've done the first part of gastrointestinal system, and I showed you slides up to stomach. And in this laboratory, I'm going to show you slides from intestine and some glands. Okay? So we'll start with duodenum. We will start with duodenum. So this is the lowest magnification of duodenum. As a general rule, as you have learned in your theoretical lectures, and as a general rule, the gastrointestinal system has four layers. I can use the mouse here. Okay, the gastrointestinal system has four layers. So this is the lumen of the organ. This white area is the lumen of the organ. And starting from here up to here, this is the mucosa. This is the mucosa. And beneath the mucosa, when I magnify the image, you will see it clearly. Beneath the mucosa, you will see the muscularis mucosa layer. You will see the muscularis mucosa layer. And this is this area where I'm showing with arrow is the submucosa. Okay, this is the submucosa, and this is the muscularis externa layer. This is the muscularis externa layer. And as you have learned in your theoretical lectures, the muscular layer of this stomach is straightened. Is it straightened? It's smooth, isn't it? Yeah? All right. So I will show you 3 slides from intestine, and I have started with duodenum. And the histological distinguish of duodenum from jejunum or ileum is to find out or to see Brunner's gland. Okay? So this area in this submucosa, you will see Brunner's glands, alkali mucus secreting glands, Brunner's glands, and these Brunner's glands found only in the duodenum. Okay? All right. Let's have a look to the mucosa in that. Another thing in intestine is plica circularis. Do you remember plica circularis? Yeah? Okay. I'll show you. As I told you, this white area is the lumen of the organ. This is the mucosa. This is the mucosa. Beneath the mucosa, there is muscularis mucosa, and this is submucosa, and these are Brunner's glands and muscularis externa, the muscle layer. And in intestine, only in intestine, duodenum, jejunum, and ileum, there's permanent foldings. There are permanent foldings. These are the permanent foldings, like this one, okay? So it's like a protrusion towards to the lumen. These are called plica circularis. These are called plica circularis. And in duodenum, ileum, and jejunum, these plicas are permanent. Okay, these plicas are permanent. For example, in large intestine, these plica structures are not permanent. Okay, but in duodenum, jejunum, and ileum, plica circularis is permanent. All right, let's magnify our image. Structures in the mucosa, these structures are villi, okay? These are villi structures. See, these individual structures are villi. And this villi is lined by simple columnar epithelium. microvilli. These simple columnar epithelium have in the apical side, they have microvilli, okay? And all these microvilli makes a brush border or straighted border. And among the straighted border, you can see these cells, which are goblet cells, okay? These are mucus secreting cells. It may seem complicated, but I'm going to repeat it one more. Listen to me very carefully. So, in duodenum, ileum, and jejunum, you have mucosal foldings. You have mucosal foldings like this big mucosal folding. This is called plica circularis. Okay, this is called plica circularis. And this plica circularis is made up of from villus. So these are villi, individual villi. When I look at the villi in depth, I'll magnify my image. When I magnify my image, see, this is the villus or villi. This is another villi. This villi is lined by columnar epithelium. See, this is the columnar epithelium's nucleus, another columnar epithelium nucleus. And in the apical side of this columnar epithelium, there is macrovillus, okay, or macrovilli to enlarge the absorption area. And among this columnar epithelium, you may also see these cells, which are goblet cells. Okay, so these are goblet cells. So when you look at the core of the villi, you will see lamina propria. This is the lamina propria. So lamina propria is composed of connective tissue cells and some immune cells like lymphocytes. Okay. So what did I tell you? I've told you that this is the mucosa, and beneath the mucosa there is muscularis mucosa. I'll show you the muscularis mucosa. But before muscularis mucosa, you see these structures. See, in the mucosa, you see these round structures. These are infoldings of epithelium. These are infoldings of epithelium, and we call them in particularly libercune cryptas. Okay, these are called libercune cryptas. So these are infoldings of the mucosa. Let me show you in depth. See, I'm still in the mucosa. These structures are infoldings of the epithelium, and in transverse section, you see these round structures. These are called libercune cryptas. Okay? So when we finish the mucosa, in a thin layer, just here, you can see muscularis mucosa. I'll try to find a better place to show you the muscularis mucosa. Okay, this muscle, very thin muscle layer, muscularis mucosa. Beneath the muscularis mucosa, we have submucosa. Alright, I'm here. This is the mucosa. I showed you the muscularis mucosa, and this is this area between the mucosa and muscularis externa. We have submucosa. This is submucosa, and in the submucosa, as one of the distinctive features of duodenum is the Brunner's gland. And Brunner's gland found where? Nerede arkadaşlar? Canlı mı? Submucosa. Yes. Can you find this Brunner's gland in jejunum? Apo söyle bakayım.
Yes.
Who said yes?
No.
cannot find it in duodenum or ileum, only in duodenum, okay? Only in duodenum and in submucosa, okay? So these are Brunner's gland. What's the function of this Brunner's gland? Why they are there? Alkaline mucose. Okay. So this alkaline mucose does what? Evet, mideden gelen asitin nötralize etmek için değil mi arkadaşlar? Alright? So we have Brunner's gland.
Hocam bir şey söyleyebilir miyim? Sirkler dikkat ederken submukozayı da aldınız mı?
Yes, it's a folding, yes. And plica circularis, I'll show you better in the second slide. Alright, and now, what we have, the last layer is muscularis externa, and internal longitudinal and external circular layer of smooth muscle. Okay, smooth muscle. Arkadaşlar hatırlayın, tüm iç organlarımızda düz kas vardı değil mi? Smooth muscle. Alright? Okay, let's go to the second slide, and the second slide is jejunum. Will I see Brunner's gland? Will I show you Brunner's gland? No. Why? Because it's only in duodenum. Yeah? Alright. So this is the lowest magnification. This is the lowest magnification, and this is jejunum. What's the distinctive feature of jejunum? How would you differentiate jejunum from duodenum or ileum? Come on, how?
Bililer daha mı şey?
Alright, in duodenum, yes, thank you. In duodenum, the distinctive feature is the Brunner's gland, and in jejunum, the distinctive feature is the villi is longer. Okay, the longest villi is found in jejunum. Look, these are villi. Okay, these are villi, and these are the long villi. The long villi found in where? Jejunum. Okay, so in this slide, the layers doesn't change. It has mucosa, it has muscularis mucosa, it has submucosa, and it has muscularis externa. Just remember this for gastrointestinal tract. But plica circularis. Are plica circularis permanent here? Is it permanent? Yes, it is permanent. Where it is not permanent? In large intestine, it is not permanent. So this is the plica circularis. Look, I'm going to draw a real big circle. See these foldings, which includes mucosa and the lamina propria, and a little bit of submucosa. Okay, so this extravasation towards to the lumen. This is plica circularis. This structure, okay? Hold on. This is a plica circularis. This is another plica circularis, and these are permanent. And plica circularis is made up of from villi. So these are villi, all right? And villi is the longest in jejunum. And when you look at villi in depth, again, it is lined by what? Columnar epithelium, isn't it? Columnar epithelium. Arkadaşlar beni takip edebiliyor musunuz? All right. Unfortunately, the image is not clear, but we can study in your individual microscopes as well. So this was a villi, this is another villi. When you look at the villi, this villi is lined by columnar epithelium. Columnar epithelium. See, this is the columnar epithelium. And these are what? Goblet cells. These are goblet cells. So when you go from intestine, from duodenum towards to the large intestine, we will always see goblet cells. But what do you think? The number of goblet cells, will they increase towards to the large intestine? Would we need more mucus or will it go down? You think it will go down? You are wrong. Yes, the number of goblet cells towards to the large intestine. When I show you the large intestine slide, you will see more goblet cells, okay? Because you need more mucus to be secreted. Alright, so this is the villi, and the core of the villi is again filled with lamina propria. And I have mentioned this, in my first slide. What are these? I am still in the mucosa, and these are again foldings of this mucosa. This mucosa foldings are libercum cryptas. Okay, libercum cryptas, and this is all lamina propria. And again, so because this was mucosa, beneath the mucosa, what do we have? We have muscularis mucosa. Okay, and inside mucosa, I'll go to the lowest magnification. So this was my mucosa. This is plica circulare. This is villi. The villi is lined by columnar epithelium, and each individual columnar epithelium has microvilli. And all these microvilli makes a brushed border or situated border. Okay? And this is the what? Submucosa. This is the up to here. This is the submucosa, and then we have muscularis externa. Okay, this is the muscularis externa layer, which is made up of from smooth muscles. All right, I'm going to the third slide, which is ileum. Ileum is a part of large intestine. Is it right? No. Uyumayın. Evet, değil. Yanlış. Okay, ileum. So, what's the feature, distinguished feature of ileum?
Biraz daha fazla mukoz var jejunuma göre.
Neler?
Biraz daha fazla mukoz yok mu jejunuma göre?
Efendim?
Yes,
Pairs patches. That's true. Pairs patches. That's true. Thank you. Remember pairs. Wow. Alkış arkadaşlar. Yes, that's true. Pairs patches. Remember lymphoid follicles found in the ileum, and in ileum it is called pairs patches. But the other feature of ileum is, yes, you are right, the number of goblet cells increase, but the villi structures is getting shorter. The villi structures is getting shorter. Okay, so this is the villi, and the core of the villi is still filled with lamina propria. And what are these things? These are enfoldings of mucosa, and these are called libercum cryptas. Okay, these structures are libercum cryptas, and these are enfoldings of mucosa. And do we have Brunner's gland here? No. This is the submucosa, and this is muscularis externa. And I'll try to show you the pairs patches or lymphoid follicles. Yes, there you go. So these pairs patches or lymphoid follicles found in the which part? Arkadaşlar gözünüz önünde, hadi. Neredeyim ben şu anda? Söyle. Submucosa. Yes, the pairs patches or the lymphoid follicles are found in the submucosa. externa, and this is submucosa. Alright? Okay. I'm in the large intestine. Okay. Someone tell me the layers of large intestine. Gökhan. Gökhan söyle bakayım large intestine'ın layer'larını. Fatma Nur düzenli. Bak bildim adını. First layer. First layer. Yes, mucosa. Second layer. Beneath the mucosa, what do we have? Ne var? Muscularis mucosa. Arkadaşlar biraz önce saydım. Uyanın. Mucosa, muscularis mucosa, beneath muscularis mucosa, what do we have? Yasin, ne varsa sende vardı. Söyle canım. Submucosa and beneath mucosa, Füko söyledi. Muscularis externa. Thank you. Alright. So, as I said, gastrointestinal system layers, there are 4 layers. It never changes. Mucosa, muscularis externa, submucosa, mucosa, muscularis mucosa, submucosa, and muscularis externa. And this is the, here, have a look. So this is the lumen of the large intestine, and this is the crypta, okay? This is the crypta circularis, the foldings of the mucosa and submucosa. And remember, what did I tell you? In duodenum, jejunum, and ileum, these foldings, these cryptas are permanent. But in large intestine, these foldings, these cryptas are not permanent, but organized by the constructions of this muscularis mucosa layer or muscularis externa layer. Okay? And the other thing, the other feature of large intestine is, have a look, see, this is the mucosa. Alright, this is the mucosa. This is muscularis mucosa. This is submucosa. Submucosa continues where? Okay, up to here. Okay, this is the submucosa. And then muscularis externa. These pink structures are muscles. And the other important feature of this large intestine is, as you may see, there are no villi structures in large intestine. There are no villi structures. Let's have a look in that. Let's magnify our image. See, this is the mucosa layer. This is the mucosa layer. This is lined by columnar epithelium again. And as you see, the number of goblet cells increased here. Okay? The number of goblet cells increased. And these crypts, these foldings of mucosa, these crypts make intestinal glands. These are all intestinal glands. Okay? These are all intestinal glands. These crypts are all intestinal glands. And there is no villi structure in the mucosa. And as I told you, this is the muscularis mucosa, and then submucosa, and muscularis externa, you may see. Alright, we have finished intestine and large intestine, and the next slide I'm going to show you is gallbladder. Gallbladder. Histologically, it's a really easy slide. What's the function of gallbladder? Storage and concentration. Storage and concentration of bowel. Okay, storage and concentration of bowel. Okay, you can see. As I said, as I've told you, it is really easy. We have mucosa, which is made up from columnar cells, but the mucosa is not regular. It is not regular. It makes foldings. like this. It is not a regular mucosa, but irregular. And it is lined by columnar epithelium again, in between goblet cells. And we have, beneath the mucosa, we have muscle layer. So these are all muscle layer. But this muscle layer is dispersed in between this connective tissue. Okay, these are all connective tissue cells. These are all connective tissue cells. And among connective tissue, you may see muscle tissue like this. These are all muscle tissue. This is connective tissue. So what I'm trying to tell you is the gallbladder, histologically, the feature of gallbladder is the mucosa is not regular. The mucosa is not regular. It is folded like this, but this is not alveoli. This is not alveoli. And beneath the mucosa, we have connective tissue. Okay, connective tissue. And among this connective tissue, you will see muscle tissue. These are all muscle, smooth muscle. Okay, smooth muscle tissue, and these are all connective tissue components. All right. Okay, we finished the gallbladder, and the next slide is liver. I think you are familiar with the liver slide. I assume we've shown you in your first year. Okay. In liver slide, I'm in the liver slide. So classical liver globule is a hexagonal in shape. Okay, the classical liver globule is hexagonal in shape. Hold on a sec. Okay, like this. See, this is a lobule, this is a liver lobule, and as I have told you, it is hexagonal in shape. And in this lobule, in this liver lobule, there are three things I want you to find out. The very first thing is the central vein. It is in the central, as I'm showing you, it is the central vein. In the periphery, in the angle, at the angle of this hexagonal shape, you will see portal striates. Here, okay, you will see portal striate. What are the components of this portal striate? Hepatic artery, portal vein, and bile duct. Okay, this makes the portal striate. And the third thing you will find in the globule is hepatocytes, the liver cells, hepatocytes. Let's try to see central vein. Okay, so this is our central vein. And starting from the central vein, the hepatocytes are arranged in rows, as I'm showing you. And these rows, see, this is a row, this is another row. These rows are separated by sinusoids. Okay, these rows are separated by sinusoids. Where can you see sinusoids apart from liver? Give me another example of sinusoids. Where can you see sinusoids apart from liver? Come on. Maybe where? Bone marrow? Yeah? Ring a bell? Yeah? Okay. So these hepatocyte rows are separated by sinusoids. And the other structure I'm going to show you is portal striate. Unfortunately, the image is not clear, but we can study in your individual microscopes. As I told you, you will see hepatic artery, portal vein, and bile duct in this portal striate. The last slide I'm going to show you is pancreas. So the feature of pancreas is functions as exocrine and endocrine organ, okay, or gland. Pancreas has endocrine and exocrine gland functions. So the part where it functions as endocrine gland is called what? What do we call the endocrine part of pancreas? Langerhans Islands. Langerhans Islands. And among this exocrine gland, I am showing you the Langerhans Islands. See these pale staining areas. These pale staining areas are the Langerhans Islands, and these are made up of. Oh, sorry. Okay. These are Langerhans Islands and endocrine part of pancreas. So in the Langerhans Island, there are individual cell types, but it is not readily identifiable in this staining. We need anticore immunohistochemistry to be able to identify these individual cells. Okay, I will show you the exocrine part of pancreas. See, I'm in the exocrine part of pancreas. So this is a pancreatic acini. This is a pancreatic acini. Another pancreatic acini. Another pancreatic acini. Okay? So in the center of this pancreatic acini, what do we have? In the center of this pancreatic acini, what do we have? Yasin, hadi söyleyeceğim.
Çok uzakta kaldım.
Centroacinar cells. Centroacinar hücre. Hatırladınız mı arkadaşlar? In the center of this pancreatic acini, we have centroacinar cells. Okay, centroacinar cells. Unfortunately, it's not clear here, but in the acini, the location where the nucleus is located stays more basophilic, and the cytoplasm stays more eosinophilic because of zymogen granules. Okay, because of zymogen granules, the upper cytoplasm stays more eosinophilic, but the lower part of this cells stays more basophilic. Alright, I would like you to distinguish all these structures that I have shown you. Please go and take your slides from our technicians, then I'll come and help you to find out all these structures. Thank you.